Talking like this, I know some people may have gotten used to it by now. When I taught in the fall, we always had students present in an in-person component. And the one time when I did have to talk without having an audience present, it felt really weird. So it feels really weird again. Hopefully I'll still manage to produce something that gets the research across. But anyway, that's just the world we're living in these days. I want to just check to make sure when I look at my screen, the slideshow here, at the very top, there's an outline bar, which shows the different components of the talk, but part of it is covered up by Zoom informing me that I'm screen sharing. I don't know if that's going through to everyone else or not, but if it is, and you can't see the outline bar at the top, then I can shrink it a little bit and adjust some things.
We can see everything.
Okay, great. Okay, so I'll just get started. I want to talk about a new mechanism for the old phenomenon of population cycles and an application to deer and deer vehicle collisions. So it is a theoretical mechanism, which nobody has noticed up until this point. And then actually the way we realized the mechanism is a mechanism is we kind of did it. We didn't approach it theoretically. We approached it empirically from the data sets. And gradually over a long period of time, we realized what was actually going on in the data, and that allowed us to frame the mechanism and then come up with a theoretical explanation for the mechanism after that. But it very much was led by the data. So I want to start, though, by just talking about population cycles, because whereas it is a really old topic of study and area of interest in ecology, it may not be in your own personal wheelhouse, so it pays to sort of remind you what that kind of stuff is. So probably the example that you're most likely to have heard of just from a basic ecology class back when you took one is this lynx hare example. And here are the time series that get trotted out usually in basic ecology classes. And the point here is that both of these things are going up and down quite regularly. Now, the height of the peaks is not necessarily regular, but the spacing of the peaks is quite regular. So there's this periodicity. I'm calling it fairly regular periodic oscillations because sometimes people argue about how periodic is periodic enough. And I'm going to just say, let's not be pedantic. That's periodic. Take a look at the time series. The amplitudes need not be consistent to be counted as a cycle. So that's one example that you're likely to have heard of. There are lab experiments that people have done that demonstrate examples. So there's this old bean weevil experiment. It's a petri dish experiment using, and the sort of main players in the experiment are beans, of course, bean weevils, and a parasitoid wasp. And Uchida observed the system, monitored it, and counted everything over a period of 6 years and got time series which looked like this. So during some, but not all, of the dynamics here, you can see what look like periodic fluctuations or semi-periodic fluctuations. That's another classic example. The Hoffaker's experiment with citrus mites is another classic example. So just as a reminder, the players in this experiment are oranges, the citrus mite, and a predator of the mite. And Hoffaker was attempting to understand what could produce predator-prey cycles, what was necessary. He noticed that it couldn't happen on just a single orange, but it could happen if you had a spatial array of oranges. And so space was a crucial part of what produced the cycle in this case. So here's the data. On the bottom is the total number of mites and parasitoids in the system of one of Hoffaker's experiments. And you can see this periodicity in both species. It didn't come about through periodicity in any of the individual oranges. So each of these rectangles up here is like a map of the field of oranges that Hoffaker used as his experimental system. And whether they're black or white indicates whether they have mites and so on them. And if you take a close look at these, you can see that no individual orange is doing anything very persistent at all. There's lots of extinction and recolonization, but if you add up across space, you get a cycle. So that's another, for the purposes of this talk, I'm just giving that as an example of a cycle that you're likely to have heard of before. Voles is another example. Vole populations in northern Scandinavia have been heavily studied, and they cycle wildly in many cases. The one I'm about to show you is from northern Finland using a gray-sided vole, and here's time series there. So you can see they go up and down quite a lot and quite regularly. So it has the same features that I've been claiming are needed, or that I'm defining are to be needed to call something a cycle. You've got this fairly regular spacing of peaks. It's not just going up and down randomly, but the peaks are regularly spaced. So there's this periodic component to it, and the amplitudes need not be consistent through time. Here's another example of a traveling wave this one makes. And the players in this case are the larch bud moth and species that it interacts with in Europe. So the larch bud moth, of course, eats the larch, and it is eaten by parasitoids. And so these cycles, I'll show you a video of them in a minute, but they've been taking place very regularly for an extremely long time in parts of the European Alps. And they've even, if you look at old timbers from really old houses, so hundreds of years old timbers, you can see, if you look at the growth rings in these timbers, you can see spacing which is consistent with a periodic defoliation event, reducing the growth rate of the trees in that year. And that can be attributed most likely to the larch. And so the oscillation of the larch has been going back centuries, if not longer. So let's look at this movie because it's cool. So what you're looking at here is a map of, this is northern Italy. So this is a map of part of Europe. And each black dot on here is a location where defoliation was monitored. And it's a movie where the blobs represent how much defoliation was going on at that point in time. And this is a sum total across all the locations which were monitored. So you can see a cycle here that went on for 3 cycles, and then it seems to have disappeared towards the end here. And if you look at any individual location on this map, you can also see that if you were just present in that location throughout the entire time period, you would see a defoliation cycle, which would correspond to a larch abundance, a bud moth abundance cycle in that location as well. And there's a traveling wave aspect to it as well. Okay, so these are a bunch of examples you've probably heard of, all to serve the common purpose of sort of motivating that it's worth studying these things. If you aren't satisfied with examples and you want to know if it's really worth studying these things from sort of a commonness viewpoint, here's a paper from 1998 where they looked at a big population dynamics database, and they found that in that database, at least, nearly 30% of the time series were cyclic in the sense that I'm talking about here. Now, of course, that population dynamics database may not have been representative of all populations. Ecologists like to study cycling, so maybe they gravitate and study things that cycle, but still there's enough of them, there's enough cycling populations to make it something worth studying. So just to summarize some of the characteristics I've said, what do I mean by a cycle? Periodic oscillations is what I mean. Okay, they don't have to be as periodic as a sine wave, it's ecology after all. Let's not be too pedantic about it, but they do tend to be pretty periodic when we're talking about cycles, and the amplitudes aren't required to be regular or consistent. Now, some of the observations based on these examples and other examples are the following. Many of these examples involved some kind of delayed density dependence, usually through trophic interactions. Like let's take the larch bud moth example. If the bud moth population grows, it defoliates the trees, which means the next year the tree needles come back and they're stunted and they're not as nutritionally valuable. So that exerts a kind of delayed density dependent effect the moth exerts on itself, because being abundant last year makes it more difficult to be abundant this year. Likewise, the parasitoids that parasitize the moth, if the moth is abundant one year, the parasitoids will become abundant, and then the next year there'll be a lot of parasitoids. It'll be hard for the moth to be abundant again. So those are a couple of trophically mediated ways where delayed density dependence can kick in and bring about cycles. It can also happen through maternal effects and other mechanisms, But the point is that most of the work that's been done on mechanisms for cycles has centered on, and at least over the last 30 or 40 years, has centered on delayed density dependence kinds of mechanisms. And what I want to do with this talk is introduce a new mechanism that we discovered by studying deer and deer vehicle collisions. So I'll show that delayed density dependence isn't strictly necessary. That's what the new mechanism is. It doesn't involve delayed density dependence. And so we're going to elaborate that mechanism. Cycles have been studied for a very long time, over 100 years, and apparently no one noticed this before. And we'll argue that in fact, it could be quite common as a mechanism. We don't know that for sure, but that it has the elements which would enable it to potentially be common. And in the process, I'll use some cool novel statistical approaches, which are the tools that we use to notice that this was going on. And I should say, before I go any further at all, that this is work by Tom Anderson, Lawrence Shepard. I think Lawrence is in the audience. Hi, Lawrence. And John Walter. Those three you probably know because they have been hanging around KBS. Tom and John have moved on and Lawrence is about to move on, but they've all been around. You may know them personally. Anyway, this is work led by them, Ecology Letters. You can read about it if you want more details. Okay, so what I want to do now that I've kind of introduced what population cycles are and where I'm going is try to give some basic intuition for the mechanism, just by talking not about complicated ecological scenarios where there's all kinds of difficult to understand things going on, but just some simple toy made up scenarios where we've stripped away as much of the complexity as possible. I just want to get across the main idea here. And then we'll go to the real systems. So the idea is I'm going to have several slides where I'll have an idea on the left and some notation for it on the right. So suppose we have a bunch of population time series in locations which I'll index by I. So those are just the sampling locations, like places where you go out and you count the populations or you measure the population density of your species of interest. And we're going to suppose that the population density has been measured in all the locations over the same period of times. And so the notation we give for that is just time series notation. P1 in location 1 as a function of time is what's been measured. Now, it turns out that any time series can be decomposed into sine waves at different periodicities. This is called a Fourier decomposition, and it's an essential tool that we're going to use. You don't really need to understand how it's done. You just need to understand that it can be done. So basically what you do is population fluctuations on different time scales or frequencies are separated for each time series of data. So we take our time series, which were denoted P sub 1, P sub 2, up to P sub n, and we break each one up. And the superscript here is intended to represent the sine wave component of this time series, which happens at different frequencies. So this is one of the frequencies. This is the same frequency, but it's a different component because it's a different component of a different time series, and so on. So we've decomposed each of our time series into sine waves, essentially. And then the sine waves on some time scales may be synchronized across locations, even while the sine waves on other time scales are not synchronized. So it may be, for instance, that all the sine waves that are a component of all of our time series on time scale 2 here. So this one, all the way down to this one, maybe all of them are synchronized, and that can happen at the same time that, let's say, all the 3 time scale components are not synchronized. So synchrony is, it turns out, time scale specific in that way. When you see that time series can be decomposed into sine waves, you can imagine how that can happen. It just comes out that you get synchrony of some of the components, but not synchrony of other components. So how can you have a bunch of sine waves of the same time scale or the same periodicity be synchronized? Well, here's the picture. Here's a bunch of sine waves of the same periodicity. Their phases are similar, and so they're synchronized. That's pretty clear, hopefully. And here's a bunch of sine waves. All of them have the same periodicity, but they've all been shifted around, right? Their phases are all different from each other. So whereas in this case, they all go up, they all peak at about the same time, they all trough at about the same time. In this case, they peak and trough at all sorts of different times. So these ones are synchronous, these ones are asynchronous or unsynchronized. So the idea here is that we add time series across space. And when you do that, since we've decomposed each of our time series, we can envision that as if we just add up the time series across space, that's the same as adding up the corresponding components. So adding up all the 1 timescale components across space, adding up all the 2 timescale components across space, etc. So adding up the components is the same as adding up the whole time series. And this will just be also a decomposition, although a different kind of decomposition, of the total time series that you get the spatial total population by adding up all your time series across space. Now the idea here, and this is where we start to get at where a cycle comes out, synchronous components add up to a powerful fluctuation. If you added all these sine waves up, because they're all reinforcing each other, what you'd end up is a massively strong sine wave because they're all working together. But if you added up all these sine waves, they would mostly cancel out because they're opposing each other at different times. So in the same way, if let's say all of the 2 components of all of your time series from all your locations are all synchronous, then when you compute this sum right here, that will give you a big fluctuation on that same time scale, that same periodicity, the time scale 2 in this notation. It's going to be a big fluctuation, but let's say P1 and P3 are a bunch of asynchronous sine waves, then those are going to add up to something small. Those will average out and disappear, and that's how you get a cycle on the synchronous time. That's how you get a cycle in the total. So what are the key features of this mechanism? It's the spatial total population that cycles. That's what we're talking about here. Like it may not, the local populations may not cycle or not noticeably cycle, but because there's a small oscillation across all your localities, and those small oscillations are all synchronous, they may be totally obscured locally by noise, but because they're synchronous, when you add everything up, all that noise cancels out, and what gets left up, what you can see in the spatial total is the components of the local time series, which are synchronous, and that produces a cycle if that synchrony occurs only on certain time scales. So it works because some time scales of oscillation are synchronous and others are not. That's the key there. Those ones that are not don't add up to much in the spatial total, but the ones that are do add up to a lot, and you get cycles on that time scale where the synchrony occurs. The synchronous time scales reinforce each other and add up to a lot. The asynchronous ones don't. That gives a cycle. And only the synchronous time scales matter in the spatial total population signal. So this quote kind of summarizes it, although obviously Helen Keller was talking about other things. Alone we can do little, together we can do much. So if you've got time scales of oscillation where all your time series are synchronous, so they're working together, that adds up to a lot. If you've got time scales of oscillation where all your time series are not oscillating together, they will conflict with each other. That will add up to very little. And the time scales that get emphasized in the spatial total are those ones that were synchronous, and that produces the cycle. So I just want to give some pictorial examples so you can get a sense of what these are. This is again made up. We'll get to the real data later, but I want to allow you to actually see some time series instead of just thinking about them. So here's a panel full of time series. I only colored them differently so you can distinguish them from each other, but that coloring doesn't mean anything. Each of these time series is a sine wave of the same frequency, but random phase shifts, plus a lot of local noise. There's way more local noise than the amplitude of the sine wave. The result being you can't tell, this just looks like a plate of spaghetti to me. The middle panel is, we're imagining these to be environmental noises taking place in a bunch of different locations. And then we're going to stimulate an autoregressive model in each location with these noises. to give us some modeled populations. So these are meant to be populations. And when we sum all of the population time series, you get diddly squat. You get this. That doesn't look like a cycle. It just looks like some noise. However, if we are going to modify this, we're going to do almost the same thing, but we're going to modify one thing. Instead of having this sine wave that's buried under local noise in each location, have random phases, we're going to adjust it so they all have the same phase. So on this slide now, they all have the same phase. You still can't see anything. This just looks like a plate of spaghetti. This just looks like a plate of spaghetti. Maybe you can convince yourself there's some synchrony here. The point is the synchrony doesn't have to be very strong, but because we've got this tiny little sine wave, and it's the same sine wave in all the locations, even though it's buried under local noise, that sine wave reinforces when we add it up and up and up across all the locations, and we get what amounts to a great big cycle in the end. So that's the only difference between these two examples, is that this tiny little sine wave is randomly phase shifted in this example. So you get not much at the end in the spatial total, but now they're all phase shifted together. They're all cooperating. You get this massive cycle. Okay, so I hope the mechanism is somewhat transparent, at least the idea of it. Let's talk about how you see it in deer data and in deer vehicle collisions data. So first I'll just whip through the kinds of data that we're talking about here. So this is after a whole bunch of cleaning, which Tom Anderson and the other co-authors had to work quite hard to do. We got data from the Wisconsin Department of Natural Resources. We did a whole bunch of work to harmonize and clean the data. And at the end of all that, it was deer abundance per year, per county in the state of Wisconsin, about 70 counties from 1981 to 2016. And I can tell you something about how those measurements were made, but let's just start from this starting point for the purposes of this talk. And then, so we also got deer vehicle collision data. And this is again per year, per county in Wisconsin from the not quite as long a time period, but mostly overlapping, 1987 to 2016. This is from the Wisconsin Department of Transportation. So this is just reported incidents of someone crashing into a deer. We also had traffic volume data, so we could correct for that. We also had a bunch of different kinds of environmental data. So I'll just talk about the ones that actually turned out to be important in the analysis, but one of them was snow depth. So this is snow depth per year per county in Wisconsin over the same time period. We had a bunch of other weather variables too. We also had climate indices, which turned out to be important. We had an El Nino index called MEI, and we had a Pacific decadal oscillation index, and we had summer and winter versions and all kinds of stuff. The winter versions turned out to be more important because that's the rough season for the deer in Wisconsin. And so it was over this period. We also had hunting data. Now, this was an index of hunter abundance per county per year, again in Wisconsin, and it's based on sales of licenses during the first part of gun hunting season. So it's not a complete count of all hunters, but it's an index of how many people went hunting that year. And it's over a slightly shorter time period. So what do we claim to be true about these data? Basically, what we claim is that the mechanism that I just introduced is represented in these data and produces a cycle at the spatial scale of the whole state of Wisconsin in these data. So we're claiming that environmental, and so let's break that down. Environmental drivers cause timescale specific synchrony in deer across the counties of Wisconsin. And that produces a cycle in the state total deer population via this mechanism that I sketched. And then, so that's the first part with the deer. And then the second part with the DVCs, the deer vehicle collisions, is timescale specific synchrony in deer produce timescale specific synchrony in deer vehicle collisions in a way that should be pretty intuitive. And that then produced via the same mechanism a cycle in state total DVC numbers via that mechanism. So how do we demonstrate this? In other words, how do we take this mechanism, which currently exists in like abstract mathematics land, and translate it into finding out whether it applies to deer and DVCs? Well, of course, we use statistics. Talk about the overall logic of our statistical argument. How do we demonstrate that this mechanism applies? Well, there's four, I've broken into 4 steps. Step 1 is first, well, of course, if we want to argue that a particular mechanism is causing a cycle, we better start by showing that there is a cycle in state total Deer numbers. Uh, and, and then subsequently, if part of the mechanism is that there's timescale specific synchrony in Deer, and that's what's causing the cycle, then we better show that there actually is timescale specific synchrony in Deer, and it better be at the same timescales as the cycle. So first, we're just kind of taking the 2 endpoints of the mechanism, right? We're showing that we want to show that A causes B. So we better show A and show B to get started, right? That's step 1 and step 2. Step 3 is to show that the synchrony caused the cycle, of course. And step 4, which is a little bit ancillary, but still important to understand where it's all coming from, is to show that environmental drivers caused the timescale specific synchrony in Deer. So environmental drivers cause the synchrony is timescale specific, so that timescale specificity produces a cycle. That's what we're going for. And then the same thing or similar thing for DVCs. First, we want to show there is a cycle in state total DVC numbers. Then we want to show there's there is timescale specific synchrony in DVCs at the same timescale as the cycles. And then we want to show that there's a causation. And then we want to show it all comes from, in this case, not from environmental drivers, at least not directly, but from Deer. So it's the environmental drivers cause timescale specific synchrony in the Deer. And then that causes timescale specific synchrony in the DVC numbers. Okay, so now we'll just go through those logical steps. And I'll remind you what each step is before we do it. So for Deer, the first step was to just show that there's a cycle in state total Deer numbers. Well, take a look. I mean, it's pretty good, kind of jumps right out at you. So this is a detrended version of state total Deer population in the state of Wisconsin. And that looks pretty periodic, at least for a lot of the time series to me. Now, we also did a lot of statistics to show that this is significantly periodic and so on, but let's not be overly pedantic about it. Most people, if you show them this, they say, yeah, that looks like a cycle to me. And if you are pedantic, I'm not criticizing you, I'm pretty pedantic. I can show you those statistics later. And if you look at the periodicity here, if you kind of carefully track time periods between peaks, it's somewhere between 3 and 7 years. So step 2, the logical step 2, if we want to show the timescale specific synchrony causes cycling, we just showed there's cycling. Now we have to show there's timescale specific synchrony. So that's step 2. Show there's timescale specific synchrony in Deer at the same timescales as the cycle. So this picture on the right here is what we call a wavelet mean field. And I'm not going to tell you, I can tell you outside of time if you want how it works. I can even give you code. There's a package on CRAN and so on. But I'll tell you now how to interpret this. Across the bottom is year of the data. Across the top is time scale. And the color intensity is basically how strong synchrony is at that time and at that time scale. So this is a method which just tells you how synchrony breaks down by time scale and by when it occurred. Warmer colors mean more synchrony. And I've got dashed lines here at time scales of 3 and 7 years. And you can see most of the synchrony that Deer and Wisconsin exhibit is between those time scales, 3 to 7 years. So now I've got the 2 endpoints of my supposed causal relationship. They have shown that they exist, and I want to actually show the causal relationship. I think in the interest of time, I'm going to skip talking about how the wavelet mean field works. I'm sorry about that, Lawrence. I know you're probably groaning right now that I'm not saying how the method works. There's just not going to be enough time. And I'm happy to return to it if someone asks me about it. So step 3 is to show that the synchrony caused the cycle. So how do we actually get at this causation? Well, remember that time series can be decomposed into sine waves. That's what we talked about when we introduced the mechanism. We used a way to randomize away that synchrony using a kind of resampling type method at certain time scales only. So basically, we randomly shuffle the phases of the oscillations in our deer time series, but only the phases of the components that occur at the time scales from 3 to 7 years, while keeping other statistical properties of the time series fixed. So it's a randomization method. If it's true that timescale-specific synchrony causes cycling in the deer, then we ought to be able to randomize away that timescale-specific synchrony, recompute the total state total using the randomized data, and the cycle should have disappeared, right? If you want to argue that A causes B, then take away A, and if B also goes away, well, that's some evidence, right? So let's do that. The black line here is what we just looked at a little while ago. It is the detrended state total Wisconsin deer population. The gray lines are the same thing, but based on these randomized data sets where we've removed the synchrony between different counties on 3 to 7 year timescales. So you can see the cycles. I mean, that was present in the plot that I showed before. These red lines just illustrate how big the cycles are. They're about up to about 250,000 deer, which is about 25% of the population. And that's much greater than what you get in the randomized versions. So basically what this shows is if you take out the synchrony at 3 to 7 year timescales, then that also makes the cycle at the state spatial scale disappear. So step 4 of the logic is to show that environmental drivers cause the timescale specific synchrony in the deer. This step was the most statistically involved, and it was the one that the referees tortured us most extensively about, like doing all sorts of due diligence and background work to double, triple, quadruple check it. I'm going to skip all that, but you can talk to us if you want to know more about it. In particular, if you're interested in applying similar things to your own data, we have our code available on CRAN, which we can show you how to use and so on. Now, in order to still get some of the sort of idea of how this step works, it's quite complicated for deer, but for DVCs, it's simpler. And the ideas are similar. So when we come to the step 4 for DVCs, I will talk about that one. Okay, so now let's go through the 4 steps for DVCs. It's basically the same 4 steps. Step 1 is show that there is a cycle in state total DVC numbers. So look at the black lines only on these plots at the right. Ignore the gray lines for now. There is in DVCs, for reasons that we haven't investigated, there's some low frequency fluctuations superimposed. So you can see this kind of long-term trend right here. Well, it's not a trend, it's a fluctuation. That, I don't know what causes that. I don't care for the purposes of this research. That is basically something happening on much slower timescales than 7 years, caused by some other factor. We took it out. So down on the bottom here, the black line is basically this black line, but with that slow fluctuation removed. And now you can see the cycles. So here you can see the cycles as these bumps on top of that long, slow fluctuation. Here you can see this, having removed that long, slow fluctuation, now you can see the cycles that we're after here. And these are DVC cycles, again, on 3 to 7 year timescales, about 3,000 or so DVCs from peak to trough. So that's the cycle. It's slightly obscured by this long, slow oscillation in the case of DVCs. So step 2 is to show that there is timescale specific synchrony in DVCs at this 3 to 7 year timescale. Well, here it is. This is the wavelet mean field again for DVCs. And you can see the synchrony is mostly in the 3 to 7 year range here. This synchrony up here is synchrony that corresponds to that long, slow oscillation that I just showed. So we've got the 2 ends of what we're claiming is a causation. We want to show that cycles cause synchrony. We showed there are cycles. We show there is synchrony on the same time scale. Now let's do the causation. We're going to do it in the same way we did for deer. We're going to do a randomization where we basically, through randomization of the data, remove the synchrony that occurs on 3 to 7 year timescales and see what that does to the state total DVC time series. So the black line is the actual state total DVC time series. The gray lines are the randomized versions of that. And so when you remove synchrony, you wipe out that cycle. So I said I would give an example in the DVC case of the statistical logic step 4. Well, here it is. In this case, it is showing that Deer synchrony is what caused DVC synchrony. I should start by saying that this isn't that hard to believe, right? These are Deer vehicle collisions, so deer are involved, and it makes sense that, you know, Deer synchrony, at least it's plausible that Deer synchrony would cause DVC synchrony. Well, let's take a look. The top panel here is the wavelet mean field for Deer. I showed you this before, I'm just reproducing it here. And the bottom panel is the wavelet mean field for DVCs. This is what I call the, if it walks like a duck and talks like a duck argument, it must be a duck. So let me break down what I mean by that. These two things look quite similar in the 3 to 7 year band. A lot of the details are similar. And so, but the key here is that there's enough detail for that similarity to actually mean something. If we didn't give a timescale specific description of synchrony here, if we just gave a single number, like let's say we characterized the strength of synchrony in deer by a number, 0.8. And let's say we found that using that same statistic, the synchrony in DVCs was 0.79. Well, that doesn't mean that deer synchrony causes DVC synchrony, because one number could be the same just by chance, right? But when you have a more detailed description of synchrony like we've got here, it is more reasonable to base an inference on the similarity here. Now, that said, that isn't the only argument, not by far, that we have that it was deer synchrony that causes DVC synchrony. It's just an accessible one. So I'm going to leave it at that. But if you want to know the actual statistical argument, it's based on something called wavelet coherence, and we'd be happy to talk to you about that. Some other observations. So that completes, I should say, that sort of completes the overall argument that this mechanism is taking place in deer and DVCs to cause the cycling in deer and DVCs at statewide spatial scales in Wisconsin. But there are a few other kind of outcomes of all this work. One is that we show that synchrony can cascade from environmental to population to human systems in a cascading manner. So that was just present in the talk. I mean, it goes from environment to deer to DVCs. And that kind of cascading Moran effect was something, as far as we know, aren't aware of any examples where synchrony has gone down to human systems in that way. So it's yet one more way that environmental factors can sort of have their impact on humans. And another observation is that if we have showed, which I think we have, that environmental synchrony causes population synchrony, which then causes cycling at the statewide spatial scale in deer, and that opens the possibility that climate change can alter synchrony and therefore alter cycling. So that's just an observation worth pointing out. We haven't showed that specifically here, but it opens that possibility. So I just want to say a few words about generality here. We don't know, like speaking purely as a conservative scientist here, we don't want to say that this is general because we've only checked in the one system, right? But it does have components which we know to be general. So we've demonstrated this new mechanism. How do we know how general it is? Well, lots of our earlier work has shown that timescale specific synchrony is a common thing. We've shown that in all sorts of different systems, marine and freshwater, terrestrial, all kinds of systems. In fact, I recently saw a demonstration of timescale specificity and synchrony of salmon populations in California, which that was new to me until yesterday. So these things keep coming up. The presence of timescale specificity and synchrony is apparently common based on the multiple different systems that we've looked for it in and found it. So given that that is at the root of the mechanism, it's at least a plausible hypothesis to think that it might be reasonably common for population cycles at large spatial scales like the state of Wisconsin to be caused by that kind of synchrony. And that's it really. Thanks for listening. I'm happy to take questions, and I just want to reemphasize, this is the paper if you want to read it. It's work of Tom Anderson, Lawrence Shepard, John Walter, Robert Rowley at the Wisconsin DNR, and me. Thanks.
Awesome. Thank you very much, Dan. All right, I'm gonna do the clappy thing, and then I'm gonna do the stop the recording thing, and we can